Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We muted the audio lines, but we welcome you to type in your questions for a speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Sharyn Farnsworth from Fujifilm Diosynth Biotechnologies.
Hi, good morning, good afternoon, good evening, wherever you happen to be. I'm Sharyn Farnsworth. I'll be presenting on insect cell culture and how commercial scale works in the insect cell culture world and provide some of the opportunities and challenges in a very CHO Centric world in terms of manufacturing molecules and biopharmaceuticals. Just real briefly, I'll introduce the company. We're Fujifilm Diosynth. We do a lot more than just make film. And here highlights some of our core technologies. We make products through microbial cell culture, whether it be CHO, NS0, insect cell culture, that sort of thing. And then, of course, the advanced therapies is kind of our emerging technologies with the viral vectors and that sort of thing at our Texas site. So I wanted to kick this off. A lot of people don't quite understand what the difference between insect cell culture is and BEVs, and they are two kind of distinct differences in how we define them. Insect cell culture is actually what we are developing. That's the actual cellular part of the culturing of the process. So these can be attachment or suspension cultures, and these cells can typically be cultured to very high passage numbers because there's no product present yet. And just to kind of highlight the two main insect cell culture cell lines that we see a lot in the industry, are your SF21s and its daughter cell, the SF9, which seems to be the primary cell line that's used for a lot of the more commercial projects that are using insect cell culture, and then the high 5 cell lines coming from, and both of these come from different moths, in the armyworm with the SF9 and SF21, and a cabbage looper from the high 5 cell lines. And then to quickly kind of define what the baculovirus expression systems are, this is actually the vector that carries your virus and your protein of interest into those insect cell cultures. And so we typically kind of use these words interchangeably in the industry. Typically we'll call it a baculovirus process, even though it's technically an insect cell culture process. And these baculoviruses, they go in and they infect the cells, and that's what results in making the product of choice. And it does also typically passively make more of those viruses in the cell culture. An interesting fun fact is that all baculoviruses can infect insect cells, obviously, but there are actually some that can produce recombinant proteins in mammalian cells, such as CHO and that sort of thing. That's not typically what we see a lot in manufacturing or in the industry, but they can, some of them can infect both types of cell lines. And there are more than 500 different types of baculoviruses. There may even be more since I looked this up last, but there are a lot of different baculoviruses out there that can be used to generate material. Some of the advantages of insect cell culture is that they are capable of producing very high concentrations of very large proteins. And I have high in quotations just because from a very CHO-centric world, high concentrations is is very different than what a lot of the CHO industry is used to, where we're looking at 5 to 10 grams per liter. Typically with insect cell culture, we're looking somewhere between 5 and 10 mgs per liter if we're lucky. But then again, with those processes and that sort of thing, you don't need as much material to go into your product or into your system. the multi-protein subunit complexes that are difficult to express in other cell lines are typically what is the main focus of insect cell culture. In some of these, I've given some examples. There's the VLPs, the virus-like particles. These are used extensively in a lot of the vaccine products. GSK and Merck uses it in their hepatitis and HPV vaccines, and that's a prophylactic VLP-based vaccine. Protein subunits, these use pieces or fragments of the protein to trigger an immune response. And Novavax uses this for their COVID-19 vaccine as well as their NanoFlu vaccines. Those are examples of protein subunits. And then these can also be used for therapeutic cancer vaccines, and therapeutic cancer vaccines are used to stimulate the patient's immune system to respond to whatever that particular cancer is. And Dendrion's Provenge is a commercial therapeutic used against prostate cancer, and that's an example of a therapeutic cancer protein or vaccine. Some of the limits of using insect cell culture, insect cell culture doesn't do a lot of post-translational modifications that you would see like in the CHO cell lines. They don't fully glycosylate proteins, so it's not really appropriate of a process to use for generating antibodies and that sort of thing. Also, there's not really any robust chemically defined media for insect cell culture, and I know a lot of the media companies might take issue with that, but one of the reasons for this is that the culture needs for the cell line tend to be very different than the culture needs once you infect it with a virus, when the cells are kind of struggling to reproduce and divide and that sort of thing. So the nutrient needs tend to be a lot different between those two stages. And we try to keep the same media between seed train and production. And so it's really hard to have something fully chemically defined that can be used for both stages of the process. And not having something that's chemically defined can lead to a lot of variations in the process over time. Whatever you're developing may be different, 10 years later when you're using very undefined media components. Determining virus titer in insect cell culture is also very complicated and can be inconsistent. A lot of times virus titers are done through some sort of growth assay, and those can be very variable over time. And a lot of times the viral stocks are unstable, or they don't have a long lifetime in storage. One of the other limitations for insect cell culture, and this is starting to change very quickly, especially in the COVID times, but there's very few commercially licensed products that use insect cell culture. So a lot of the commercialization roadmap pieces are a lot less well defined, and it's a little more of a struggle because you're kind of paving the way to commercialization in a lot of these processes. And then, to add on to that, the large scale data and optimization into manufacturing is minimal, if there's any of that information at all, in a lot of the journal articles. And a lot of the journal articles are still very academic, and they don't really focus on going into manufacturing or commercialization or that sort of stuff. So that can sometimes be a limitation when you're entering the market with insect cell culture. And just for making the product, there's a lot of steps. Some of these are very similar to what you would see in a CHO process. You still have to have a seed train, you still have to have a production stage, and you still have to have, a recovery sort of stage, that sort of thing. And so some of the key process steps that are a little bit different are how you will manage your viral stock, since that's where your product is actually located. How you manage that is pretty important. There are several different ways to manage your viral stock, whether it be through using BICs, which seems to be a kind of a new emerging technology that we've been seeing, and then how you scale up your viral stock, whether you're using a primary viral stock, a secondary viral stock. My preference is always to make your viral stock made to order, since it tends to be unstable over time. And then sometimes us as a CMO will see that the viral bulk is supplied to us by a third party, and sometimes that gets a little bit difficult to manage from a CMO perspective. Your infection strategy, you have to determine your infection cell density as well as the MOI, which is how many viral particles per cell that you will get into your process. A lot of times the development of this, they go hand in hand. They need to be evaluated and developed together, and that can sometimes lead to a significant amount of work. And then added that to the instability of your viral stock potentially, then it can get very complicated to develop an infection strategy and that sort of thing. And then if the process is reliant on the primary or secondary viral infection, that's an additional thing you have to look at in order to develop your process. Some of the key process steps continuing here, when you're looking at the infection cell density, just for a visual, as some people have not seen how infection cell or insect cell cultures are different from CHO processes. This is some of the DOEs that you would look at. So the pink line on the bottom would rely more on your primary infection, and basically that's you just load a whole lot of viral components into the infection, and it kills off the cells pretty quickly. Your secondary infection would be the darker purple line, and that allows the cells to grow a little bit so that more viral particles are being made while you're also making product and that sort of thing. So you'll see a little bit of growth. And then the top blue line there, that's your standard kind of CHO process that you would see, where it just kind of grows and dies over a 14-day culture. Also, you can see a big difference is that, the 6 days or so versus the 14 days of the process. So the processing time is a lot different. But there's a lot of things that go on before you actually get to production stage in insect cell culture that can also, change your time in plant. Further looking into the infection cell density and your MOI, again, kind of highlighting that the primary viral stock versus your secondary viral stock, and how your MOI and your infection cell density really play a role into that. If you have a low to medium infection cell density and a lower MOI, you may need a lot less viral stock. But then you have to generate all of that through a PVS and that sort of stuff. And high infection cell densities and high MOIs, it requires a lot more virus. And so then there will be several levels of viral stock expansion in order to be able to accommodate that sort of system. And so a lot of this just plays into how are you going to manage your viral stock, and that, is something kind of a long-term high-level vision of how this is going to play in manufacturing, is how you're going to manage that viral stock. So that becomes a very important factor when you're scaling these processes to start looking into GMP manufacturing. And again, to highlight why the media still remains the most variable part of a lot of these processes, that chemically defined media, we're really trying to partner with some of our media vendors. Irvine specifically, since that's one of our sister sites, we're really working with them to see how close to a fully chemically defined media we can get. And that just helps us to be able to have a more predictable and more robust process, for the entire life cycle of the project. Most of the media currently has some sort of yeast elate or yeast extract, and anyone who's had to use those knows exactly how variable those can be. And one thing, we do have a commercial process that we've been running for 15 years, and you can definitely see a little bit of difference due to those components over that full 15-year life cycle of the process. So that's just something to keep in mind and keep management on throughout the life cycle of your project. And a lot of work can go into determining what that appropriate media is. Some companies will change media at the production stage or add media components and that sort of thing. And one thing I always recommend is to get a custom part number for your media, even if it is considered off the shelf, and that just allows you to have a little bit more control and management over that media over the life cycle. So if there's some sort of change in, yeast delayed or yeast extract, you have a lot more control and management over that with the media vendors if you have your own custom part numbers. And then the last step that we focus on a lot is when and how to harvest. Insect cell culture, there's a lot of different ways to determine when to harvest. A lot of literature out there will say find whatever your peak cell density or your peak viability is, and once it starts declining, harvest within 24 hours. Sometimes there's a lot more work that goes into specifically looking at the protein in order to determine when to harvest, but it's not as simple as just looking at, day 14, it's starting to decline and your product quality is changing or your titer's not changing, that sort of thing, like you would see with a CHO process. So sometimes it's a little bit more complicated to determine when to harvest. As a CMO and having a few commercial insect cell culture processes, it's always preferable to have a day of harvest similar to like what you would see with a CHO, as opposed to, harvesting when your viability hits a certain percent viability, and that's just so that it's more predictable and you're more prepared for time and plant, that sort of stuff for that harvest. In different ways to harvest, obviously it depends on if the protein is intracellular or extracellularly excreted, but most insect cell culture processes do have some sort of flocculation step, because you do have that virus going in there and basically exploding the cells for the most part. And so having some sort of flocculation step to help with the recovery step is typically seen in most insect cell culture processes. And then using centrifugation versus filtration, there is a lot of depth filtration that's used in our processes. If it's an excreted product, and our preference obviously is to use the depth filtration. But again, with a lot of cell debris and that sort of thing in the process, sometimes that's a little bit more difficult, and then centrifugation becomes the preferred mechanism for harvest on that. And with that, I'm happy to take any questions. I think we have a lot of experience here at Fujifilm with insect cell culture processes and the commercialization of them, and it's always an adventure to take clients through this process, which is a little bit more different than your standard CHO processes going into GMP manufacturing. So with that, I'll take any questions.
Okay, great. So the first question is, why do you see the different growth parameters when using primary or secondary baculovirus if infecting at the same MOI?
So typically, if you are relying on a primary infection strategy for your product, you do typically have a higher MOI, and that's just because you just want to get as much virus into the cells as possible, have the cells make their product, and then harvest right then. The secondary infections usually take a little bit longer, and if you have a high MOI with that, you'll kill the cells faster than the cells can make more of that virus. So typically the MOIs change a little bit if you're relying on your primary versus secondary. But then again, if you do have a similar MOI, it's just a time in production is all that's changing. So that's kind of why those would show as different.
Okay. And how does viral titer variation impact the MOI calculations, and is there any way to mitigate this risk?
That's a very loaded question, but yes, they can be very impactful. a lot of times depending on how you're determining your virus titer, if it overestimates or underestimates what your viral load is, it can significantly impact, even though you're calculating the same MOI by calculation, what really may be there may be different than what your viral titer assay is saying. And so then that can significantly impact the process in that you're either under-infecting or over-infecting the process. And so that leads to a lot of variability in the processes as well.
Okay, it looks like we have time for just one more question. If we haven't gotten to your question, we'll be sending this to Sharyn and she can follow up with you directly. But please continue to type in your questions if you still have them. So the last question for the webcast is, how does culture variability impact harvest clarification?
So culture variability can impact your harvest pretty significantly in that if you've got a more infective culture, it could, and this is more along with the excreted proteins, but it could potentially, kill the cells off more, and so you've got more of a murky process in there, and then your throughput will be a lot less than it would be than what you've basically established with most of your development work. And so the culture variability does play a very significant role in how you harvest and what is in the soup as you're trying to harvest and that sort of thing.
Okay, well, thank you, Sharyn.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Bye.
Bye.